Light relief here. I'll talk a little bit about inferring genetic ancestry, what it's useful for, and the two main applications I'll talk about are the importance of ancestry in understanding disease and in understanding evolution. So the main problem that I'm interested in is interpreting genetic variation. So given our genome, we'd like to be able to understand what are the sets of variants that are associated with a specific disease. How do you go from changes in our genome to changes in risk for diseases? So the successful, or one of the most successful ways of doing this that has emerged over the last decade is what's called an association study. So this was something that Jonathan talked about. So you have individuals' genomes, and I guess this doesn't point. Doesn't point? No. Okay. All right. Oh, I can use the mouse. Yeah. Okay. So you have individual genomes. So you have genomes from people who have disease and normal individuals, and every one of these columns here is a place where there is a single mutation across these individuals. We call those SNPs. And GWAS, or genome-wide association studies, are a very simple study design. You look for an association or a correlation between a SNP and disease versus healthy state, right? So in this example, the first SNP appears to be perfectly correlated with your disease state, and so you would say that this SNP is associated with the disease. Of course, the catch here is these kinds of studies are subject to confounding factors, several of which are unobserved, and a very common confounder is ancestry. So the simplest setting is you could imagine all of your people who have a disease come from one population, in this case the blue population, and everybody who is normal comes from another population. So in this case, the fact that you're from the blue or the red population completely explains this association, right? So given that you are from you have individual samples from these populations, this SNP is no longer considered associated with the outcome. Of course, this is not the only kind of confounder. So we had a talk on cell type composition. That's another kind of confounder that affects different genomic assays. So clearly, this kind of ancestry can be important to correct for. Genetic ancestry, on the other hand, is also important. It can serve as a source of signal. So it can serve as a biomarker in being able to predict certain classes of diseases. So this was an example study which looked at children who have a particularly severe kind of cancer. And what this study showed was it looked at the risk of relapse for these individuals. So these children come in and they undergo rounds of chemotherapy. And sometimes the chemotherapy is not successful, so there is some risk for relapse. And it looked at what was this risk of relapse as a function of the genetic ancestry of these individuals. So basically, it partitioned these individuals into people who have low amounts of Native American ancestry versus high amounts of Native American ancestry. And what it showed was high Native American ancestry was associated with increased relapse. So this was a setting where your genetic ancestry is a biomarker. It can be used to determine treatment decisions. So now the question is, how do we actually go about inferring or understanding this ancestry? It turns out genetic ancestry is not a simple concept. It's not everyone belongs to one of a discrete set of populations. And this is due to a phenomenon called admixture. So here is one of these first studies that looked at genetic ancestry. So it looked at a set of individuals. So every column here is a genome sequence from an individual from different populations. And what it ran was a particular kind of clustering model. And what you find are there are certain individuals who belong to one cluster. So they have all of their genomes belonging to either the red or the green cluster. And then you have individuals who are mixtures of these different clusters. So that's what we call an admixed population. And just as a side remark, the underlying model that actually computes this is something that's familiar to people who are not in genetics. So for those of you who work in text, these are basically topic models, or LDA. So that was the output from a topic model run on these kinds of data. So what is admixture? So the basic process that's happening is you have a single population that splits at some point in the past. These two populations separate from each other, so their genomes diverge. And then there is some other point later on when these two populations come back together and mix. Right? And so the resulting population is what we call an admixed population. And what has emerged from the last 10 years of genomic analyses is that this kind of mixture between diverse populations is not something that's just a recent phenomenon in human evolution. So for example, if you look at recent human history, there are clear examples of admixture events. So populations like African Americans, Latinos, where we have historical evidence, where there were these populations that migrated from America into the from Africa into America. But as you go further back in past, we have genetic evidence that there was admixture across different populations. So there was admixture in the history of Europeans, so they're not a homogeneous population. And as we go further back into the past, there was admixture between humans and our closest evolutionary cousins like the Neanderthals. And so what I'm going to talk about today are a series of works that we've been involved in, where we are looking at different kinds of data that serve to inform us about these different admixture events. So let's just spend some time reviewing basic genetics. So Leonid already talked about the basic processes underlying genetic transmission. So the basic process here is recombination that's associated with meiosis. So in this example, we have a population at the top. So that's the founder population. And so every individual here has two vertical lines that's corresponding to the genome you inherit from dad and mom. And in this case, there are two populations, so people from the blue and people from red. And they produce offspring, who in turn produce offspring, and so on. So then what happens is you have individuals like this one. So this individual inherits part of their genome. So the individual on the right transmits their red genome down to the individual in the next generation. So in that case, there's been no recombination. On the other hand, if you look at these two individuals, the genome that's transmitted down is partly red and partly blue. And the reason for that is there was a recombination event. So you have a prefix which is red that shuffles up with a suffix that's blue, and that's what gets transmitted. Now, this is a random process. And so the number of recombinations is not determined. So you'll have different individuals with different numbers of recombinations in every generation. And to a first order, you can think of it as a Poisson process. So this happens in every generation. And you end up with your present day set of individuals, your admixed individuals. And these admixed individuals will have their genomes with this mosaic structure, where parts of their genomes are inherited from the blue population and parts from the red population. So the key thing is the ancestry of this individual changes as we move along the genome. And so this is a concept that we call local ancestry. So it's local because this individual has ancestry of red and blue at this position, but if you move to a different position, it'll have a different set of ancestries. All right, so what I'm going to talk about is how do we do inference about these kinds of ancestries at different time scales, informed by different kinds of data sets. So in the first part of the talk, I'll talk about some fairly old work. In fact, this was some of the early projects that got me into this field on inferring continental ancestry. So ancestry in populations like African Americans, Latinos, and so on. Then I'll talk about more recent work where we are inferring finer scale ancestry within European populations. And finally, I'll talk about archaic ancestry which is across human populations and closely related species. So here's the setup. We have genotype data from a set of individuals. So every row here is an individual. Every column here is a SNP. And for convenience, we often represent the number of alleles, the number of mutations that an individual carries at a SNP. And for most places in the genome, you only have one of two possible mutations or alleles. So you can represent each individual's genotype as this 0, 1, 2 vector. So that's the input. And what we'd like from here is basically a coloring or a painting of this individual's ancestry. And that is the inference that we're trying to make. So in this case, we're saying that the first half of this individual's genome comes from the red population, and the second half comes from the blue population. So how do you actually go about doing this inference? So typically, in many settings, we have genomes or genotypes from ancestral populations. So in this case, we have the middle part, which is the genome of the admixed population. And then we have information that tells us, for example, at this SNP, the allele 1 has 45% frequency in the blue population and 55% frequency in the red population. So that's the additional information that we have. And typically, we have this information for all of the SNPs that are available to us. So given this kind of information, now we want to segment or color or infer the local ancestry for this individual. So in this case, under some assumptions, we can start assigning local ancestry to this individual. And so we might guess that on the left, this individual has ancestry from the red population, and then there's a switch, and on the right, it moves to the blue population. So that is a very simple way of doing this. So the key complexity is these switches between the different ancestries is not known. So you have to actually infer where these switches occur. And I won't get into too many details, but the way we do this is we model the recombination process. So we say that there is recombination, and this recombination occurs at a certain rate. And given some information about how many generations this process has been occurring and what are the frequencies of the two ancestries, we can infer how likely it is to switch between the different ancestries. A second complication is often these allele frequencies. So what is the frequency of the one allele in the red or the blue population is either not known or it's noisy. And so in that case, what we try to do is we try to learn the allele frequencies from the admixed population itself using an unsupervised learning approach. So what we do specifically is we do some kind of spectral clustering within windows along the genome, and that tells us what are the potential ancestral allele frequencies across every window along these populations. And so this was implemented in this software program that we call LAMP. And the third complication is the SNPs that we see along the genome, they're not all independent. So there is correlation amongst these SNPs, and that needs to be modeled when we are making these inferences. So now I'll tell you a little bit about how we model that, and that's an extension to LAMP, which is called LAMP-LD. And the methods are described in these papers that we published a while back. And this is a problem that's been worked on intensely, and there's been many other methods that have been published since. So what we do in LAMP-LD is it's a two-level hidden Markov model, where you have one level which describes the local ancestry, and the second level describes the correlation structure within each ancestry. So what we have here is the correlation structure within the red ancestry, the green and the blue ancestries. So each of these is a hidden Markov model. And then you switch across these hidden Markov models as you switch your ancestries. So here's an example where you would trace a path through this two-level hidden Markov model, where you choose the local ancestry states. So here you would choose green, and accordingly you emit the genotype here. So in this case, you would emit this set of genotypes. Then you switch, and you then emit from this blue local ancestry, and finally you again switch back and emit from the green local ancestry. So the inference problem is we don't know which local ancestry states are associated with each of these genotypes, and that's what we'd like to compute. And this is a standard hidden Markov model computation. So just quickly go over the results. Clearly, these are outdated. There are many methods that have been developed since. But at that time, we found that LAMP-LD attained substantially lower errors on Mexican and Puerto Rican simulated admixtures compared to other methods at the time, state-of-the-art methods at that time. We then applied it to data from these two populations. So these are Mexican and Puerto Rican genotype datasets sampled from different cities in the US, and the Puerto Ricans from different cities in Puerto Rico and New York. And what we identify is that there is different proportions of ancestries in these different populations. So for example, we find that in general there is higher European ancestry in the Puerto Rican populations compared to the Mexicans. But even within the Mexicans, there is differences in the proportion of European ancestries depending on where they're sampled from. So what this is telling us is there's substantial heterogeneity even within a given admixed population. Okay, so once you have inferred these ancestries, what can we do with them? So as we said, one of these things that these ancestries are useful for is in localizing disease genes. And one strategy that has been very successful for doing this is a strategy called admixture mapping. So what does admixture mapping try to do? So once you have these local ancestries, you can compute these local ancestries in cases and controls, and then you can look for positions in the genome where there is a substantial deviation in the ancestry across cases versus controls. So the point being, there is an enrichment or an association of one of these ancestries with your disease status. So several groups have used our methods for doing this, and here is just one such example. So this is from a group at UCSF who looked at asthma in Latinos. And what they found was this association on chromosome 6 between asthma status and local ancestry. And this is just one of several examples where these kinds of local ancestry estimates have been used to map disease genes for several diseases, complex diseases in these admixed populations. Okay, so now I'll move to more recent work where we are looking not at ancestry that is continental, so not across widely separated ancestral groups, but at a finer scale. And so this is all inspired by the fact that we have high dimensional genotype data. So if you have millions of SNPs, it has been shown that you can now place or locate individuals down to hundreds of kilometers. And so one of the papers that made this point very clearly was this paper, which looked at genotypes from European populations sampled from different countries in Europe. And what they found was, by running a principal components analysis and by doing some transformations, they could actually locate individuals according to their country of origin. So essentially, if you squint, you could see that you can recapture the map of Europe based on the first two principal components. So this was based on data that was still quite limited. And now we have data sets like the UK Biobank. So this UK Biobank data set is a data set of about 500,000 individuals sampled from within UK. They are measured at about 1 million SNPs, and they've been imputed to close to 10 million SNPs. And what this allows us is an ability to ask very specific questions about the ancestry of individuals within the UK. So one thing that we would like to do is to be able to run principal components analyses on the UK Biobank data. There are a couple of challenges. First one is computational. So the typical way of running PCA is to compute an SVD. For these kinds of data sets, turns out that's impractical. There have been some randomized approximation algorithms that allow us to compute the top few principal components. There's a second issue, however, with even those kinds of approaches, and that is the fact that principal components is a procedure. What it lacks is a statistical interpretation. So what is the model actually trying to solve? And this becomes particularly important when our genotypes have missing data. And so having a way of handling missing data is important. And when we are trying to actually model correlation structure amongst SNPs. So now it turns out that people have actually thought of this issue of a statistical model for PCA. And so this goes back to the Tipping Bishop and Roweis et al. Work, where they actually had a probabilistic interpretation of PCA. So in this model, you have a fully generative model for PCA, where you have a latent variable for every individual. This corresponds to their low dimensional representation. And then you have conditioned on this latent variable, you're generating their data. So in our case, each of the n individuals' genotypes would be generated as a function of this low dimensional representation. And so this is the specific model description where the latent variable comes from an isotropic Gaussian, and then conditioned on this isotropic Gaussian, the data itself would be generated as a linear transformation with some noise. So why is this useful from an inference point of view? The reason is, once you have this latent variable model, now you can do EM for inference. So the key parameters that we're trying to infer are the W vector, the principal components, and the noise. And so now you can write down EM updates, which turn out to be computationally simpler. And so these updates look fairly straightforward. So these are the updates for the setting where the noise associated with PCA is 0. And so it turns out this is an iterative process where we are iterating between inferring Z, which are the loadings of the PCA, and inferring W, which are the principal components. And each of these, in theory, can be computed in time that's linear in the number of SNPs, the number of individuals, and the number of principal components. So if k is small, if you're interested in a small number of principal components, which is often the case in these kinds of analyses, each of these updates is actually extremely efficient. Now it turns out there's something we can do even better. So using the fact that these genotypes are 0, 1, 2, all of these iterations involve multiplying this 0, 1, 2 matrix with a random vector that changes across iterations. And so there's been recent work that has shown how these kinds of matrix vector multiplications can be performed even more efficiently. So you end up with an algorithm that's essentially sublinear as the data set scales. So we did some experiments to show whether this was an accurate algorithm. And so what we did was we varied the divergence of the data. So as the divergence becomes smaller, it becomes harder to estimate principal components. And for computing, say, 5 or 10 principal components, we measure how much the inferred principal components match with the true principal components. And across this range of values, these are very close, so it's extremely accurate. It's also very efficient. So clearly SVD, which is this blue line, doesn't scale very well. There are several other methods, which are these randomized algorithms that also scale. However, this implementation of ours, which we call ProPCA, that scales even better than these other implementations. So now we applied it to the UK Biobank. Turns out we can compute the top 5 principal components on about 500,000 individuals in about 30 minutes on standard hardware. And it's hard to see. So what we have done here is we've taken these top 5 principal components, and UK Biobank also gives us GPS coordinates for each of these individuals. So now you can plot the principal components along with the locations of these individuals. And it's a little hard to see here, but what we find is the principal component scores actually track with the geographic locations of these individuals. For example, PC1 is a PC that separates the northern England and Wales from other parts of the UK. PC2 appears to be a Wales-specific principal components, and so on. So what can we do once we have computed these principal components? One interesting idea that we thought of was we can use it to understand evolution that has been happening very recently in the UK. And the intuition is we are going to scan for SNPs which are not well described by the principal components. So now we have a Manhattan plot which has a p-value which tells us whether a SNP is well described by a principal component. And of course, there can be many reasons why a SNP is not well described. And after we filter out these artifacts, we get a subset of SNPs which seem to have no other explanation apart from the fact that these are potential candidates for selection. Some of these are known candidates. So this includes genes like TLR, which is a known immune-related gene, lactase, which is known to be under selection in Europe over the last thousands of years, HLA, which is another immune-related gene, and FUT2, which is a blood pressure-related gene. So these are known signals, but we also discover novel signals. So one of them that's particularly interesting is this gene HERC2, which is known to be one of the genes that is associated with blue eye color. And then we have another gene, which is this one, which has a non-synonymous mutation that is associated with some kind of ciliopathies. All right. So now in the final section, I'll talk about a different scale of ancestry. I'll talk about ancestry which is much more ancient. And this is again driven by a different kind of data. So this is data that is coming from ancient genomes, genomes of individuals who have lived thousands, tens of thousands, even hundreds of thousands of years ago. And over the last 5 years, there's been a major progress in our ability to extract DNA from these ancient bones and to be able to sequence them. And so this is just a review paper which shows what are all the available ancient genomes across different time points. So this is at hundreds of years old, thousands of years, tens of thousands, and hundreds of thousands of years. And what this is allowed to do is ask us questions about evolutionary processes which wouldn't have been apparent with present-day genomes alone. So here is one problem that I was particularly interested in, and this was when the first major ancient genome was sequenced from our evolutionary cousins, the Neanderthals. So just to give you a little bit of a primer, what we have observed from genetic analyses is that modern humans originated in Africa, and about 100,000 years ago there was an exit out of Africa, and that exit resulted in colonization of the rest of the world. Now, at around the same time, there was this other human species, the Neanderthals, that was present in much of Europe and East Asia. And so there's been all of this debate in evolutionary biology about what was the reason why the Neanderthals went extinct, whether there was any kind of interbreeding between humans and Neanderthals. So this was the cave in Croatia where the first Neanderthal genome was sequenced, and these were the bones. So there were 3 bones from which the first genome was sequenced. And what was observed was when they took the Neanderthal genome and they compared it to different human populations, they found that systematically the Neanderthal was closer to non-African genomes compared to African genomes. So this was a genetic relationship that indicated that there could have been interbreeding between Neanderthals and some of these human populations. So one thing that we were interested in is whether we could actually show definitively that there was interbreeding. And to do this, we looked at the lengths of these segments of Neanderthal DNA that are inherited in non-Africans. And the reason this is interesting is if recombination happens as a Poisson process, then you can think of it as a clock. And by looking at the length distribution of segments of Neanderthal DNA, we can estimate the time when this interbreeding event happened. So for example, if the interbreeding event happened just a couple of generations back, we'd have long stretches of Neanderthal DNA. If it was much more ancient, this would be broken up into shorter segments. So what we observed was when we looked at the length distribution of these segments, we could date this interbreeding event to be about 50,000 years ago. And then we did some additional analysis to show that about 2% of the genomes of non-African individuals comes from this population. So one question that we were interested in is there some kind of functional impact for this Neanderthal DNA? And so I was involved in this collaboration where we were looking at type 2 diabetes in Mexican American individuals. And this project found a new risk variant for type 2 diabetes. And when we looked at the frequency of this risk variant, we found that this variant was essentially absent in Africa. It was present in low frequencies in East Asia and at high frequencies in the Americas. And this was exactly the kind of signature you would expect if this was introgressed from Neanderthals. And what we found is when we compared the risk variant, we found that this essentially matched the Neanderthal genome. So this was very strong evidence that this was a risk variant for type 2 diabetes introgressing into modern humans from Neanderthals. So then we went about not just looking at one single locus, but trying to get a genome-wide map of where in the genome we have Neanderthal variants. And so essentially to do this, we looked at European genomes, and we compared it to genomes from the Neanderthals and from Africans. And our goal was we wanted to label every one of these SNPs as being either of Neanderthal ancestry or of modern human ancestry. So to do this, we built a probabilistic model. So one difference in trying to do this with old ancestries compared to much more recent ancestries is that the process is not Markovian. So we couldn't use a hidden Markov model. So we used a generalization which we call a conditional random field. So this allows us to account for non-Markovian interactions. And without getting into the details, essentially what we do here is we are trying to infer these hidden labels, which are whether you have Neanderthal or modern human ancestry, and by coupling it to different features that we compute from the data. So the conditional random field computes the probability of the Neanderthal ancestry given genetic data and a set of parameters. And these parameters have different interpretations. So one class of parameters, the thetas here, they couple each of these hidden labels, whether or not you have Neanderthal ancestry, to the data. And then another set of parameters, the lambdas here, couple adjacent labels along the genome. So this allows us to share information across different sites when we are making these predictions. And the key thing, the nice thing about this is this model is completely convex. So we can actually infer these parameters with standard convex optimization software. So now we actually applied this to data from the 1000 Genomes project, and we can build these maps of Neanderthal ancestry. So here is an example of the output from one of these inferences. So what we have here are maps of Neanderthal ancestry in red, which is in European populations, and in green we have the same maps in East Asian populations. So what is interesting about these maps is that there is substantial variation in Neanderthal ancestry across the genome. So there are places where there seems to be elevated proportions of Neanderthal ancestry, and other places where there seems to be depletion in Neanderthal ancestry. So what we sought to do is try to understand what might cause this variation. Is this just random, or is there some kind of a biological explanation for some of this variation? So for example, here is one extreme region in the genome. So this is a gene called BNC2. It's a gene that's been associated with skin color and freckling. And this is a place where more than 50% of Europeans today carry the Neanderthal variant, compared to about 2% who would have carried it about 50,000 years ago. And what we can show based on some amount of population genetic modeling is these kinds of regions are unlikely to have risen to these high frequencies just by chance. And what is more likely is that there was positive selection on the Neanderthal alleles. So that is the regions which are peaks of Neanderthal ancestry. On the other hand, there are actually places where there is a depletion. And what we find genome-wide is in many places in the genome which are under high selective constraint, for example, close to genes, there is actually a depletion of Neanderthal ancestry in Europeans and East Asians. And this is consistent with the fact that these are deleterious. And there is a debate on what was the mechanism that led to this deleterious consequence. So another question that was interesting, having shown that there is a deleterious consequence of Neanderthal alleles, is what might the phenotypes be that are associated with this. And so this is very much ongoing work, where we went back to the UK Biobank, where we have large sample sizes, and we can actually say what the Neanderthal alleles are in this population, and we can associate it with different phenotypes. And one of the questions we asked is, are there specific phenotypes which have high or low heritability associated with these alleles? And a general observation is for most phenotypes that we see, there is less heritability than you'd expect. And that is very likely due to, again, these alleles being deleterious. What is more interesting is there are a subset of phenotypes, lung and eye function and sleep patterns, which have elevated Neanderthal heritability. So finally, one question is, what if we don't have any of these ancient genomes? Are we constrained by the fact that we can say some of this only because we have these ancient genomes to start with? And so in, again, more recent work, we've been working on methods that try to do these inferences without access to these reference ancient genomes. And so what we try to do is we look at statistics that are computed from present-day individuals and use those to build these inferences about ancient populations. And in one such example, we applied this method to populations in Africa. So this is a setting where we don't have any of these ancient genomes. It's technically hard to access ancient genomes in Africa. And we find that there is evidence for a substantial percentage of an unknown archaic population introgressing into the present-day West African Yoruba population. And so additional population genetic modeling says that the model looks something like this, where you had a modern human population that split into the present-day modern humans and into Neanderthals. And then there was this unknown population that comes back and mixes into the ancestors of the present-day African populations. All right, so just to summarize, I think genetic ancestry is extremely useful, both for disease gene localization as well as in studies of evolution. And one of the reasons it's bound to be increasingly useful is the fact that current genetic studies have mainly focused on populations of European ancestry. So this was a paper from several years back which suggested that most of what we know about genetics comes from European populations. So this has some serious consequences about both basic biology and our ability to generalize from these studies when we are looking at other populations. So there's increasing need for studying non-European populations, and with that we have to grapple again with making inferences about ancestries. And the other important thing here is there are these new data sources, so data from ancient DNA, data from the UK Biobank, and each of these has some very interesting methodological challenges. So with the UK Biobank, scalable algorithms are a major challenge. With ancient DNA, algorithms that can account for all of the intricacies and errors in these genome sequences are a major challenge. With that, I'd like to thank several of the collaborators. All of this was done with collaborators who are present here. The PCA work was done with a very talented undergrad student in my group, and Alec, who's probably here somewhere, and Min, who's a master's student. Chris Robles worked on the Neanderthals in UK Biobank, and Arun is working on introgression in Africa. With that, happy to take questions.